Presents autoimmune destruction of myelin, MS, multiple sclerosis. Now my favorite slide of all, the slide 29, the collections of nervous tissue slide. What's the name for a bundle of axons in the central nervous system? Don't all answer at once. Bundle of axons in the central nervous system. Tract. What's the same thing if you're in the peripheral nervous system? Nerve. What's a group of neuron cell bodies in the central nervous system? That's a nucleus, and the same thing in the peripheral nervous system? That's a ganglia. Good. White matter is white because of what? Myelin. Okay? So wherever you have myelinated axons, you'll have white matter. When you don't have myelin, they call that gray matter. That's going to be either the dendrite part, the cell body part, or just remember, there are some axons that are naturally unmyelinated. Okay? Where do you find the white matter in the spinal cord? Inside or outside? Outside. Where do you find the gray matter? Inside. What about with the brain? Where do you find most of the gray matter with the brain? Outside. On the outside, right? In that thin layer that's called the cerebral cortex. Then the white matter is mostly on the inside, unless you go then really deep, and then there's more gray matter called the basal nuclei, right? All right, we then talked about electrical signaling in neurons. True or false, neurons are the only cells that are electrically excitable? False. You already learned about muscle cells, right? They're electrically excitable too. But neurons are probably more famous for it, right? That's kind of their thing. Now, before you can even start to do the electrical signaling in neurons, the neurons have to be ready for it. And when a neuron's at rest, what's that electrical difference called that you have across the cell membrane? Resting membrane potential. The resting membrane potential, and what was the value I gave you for the resting membrane potential? -70, with where being slightly negative compared to where?
Inside.
The inside is slightly negative compared with the outside of the neuron. Okay, so that's where a neuron is when it's not doing anything. Now, a big part of neuron electrical signaling is simply opening and closing different ion channels and allowing ions to either go into or out of the neuron. And there were 4 types of what are called gated ion channels. So first of all, what's the word gated mean? It just means it has like a little door on it, or has a gate on it that can open and close, right? And when you open that gate, it allows the ion to go through. When you close it, it doesn't allow it to go through. The question is, what makes the gates open? And that's what gives you the 4 different types. What causes the gates to open with leak channels? It's just random, right? It's just spontaneous. What causes voltage-gated channels to open? I went out of order, yeah. Voltage-gated channels due to electrical changes in the membrane potential, right? Just because you start at -70 for resting, that doesn't mean you'll stay there, right? That'll change, and if it changes enough, it could cause a voltage-gated channel to open. Mechanically gated channels open due to some physical pressure, vibration, touch, tissue stretching, something like that. And then lagging-gated channels are the ones that open and close when a specific chemical, like a neurotransmitter or a hormone, attaches to it and causes it to open. The resting membrane potential is negative on the inside compared with the outside. 3 reasons for why it's negative on the inside. What were the 3 reasons?
Sodium
So some of it has to do with the unequal distribution of ions because you have more potassium leak channels than you have sodium leak channels. So more potassium is constantly leaving than is sodium coming in, so that creates sort of a situation where the inside would be negative. 2 other reasons though. Anions that are stuck on the inside because they're usually proteins and they can't get out. And then 1 more. Sodium potassium pump. Don't forget that guy. That pump is working 24/7. It's like on all body cells, and that pump, how many sodiums does it pump?
3 sodium
3 out. And how many
2
potassiums?
2 potassium.
2 potassiums in. Because that's not an even, because you're not also bringing 3 potassiums in, it creates a discrepancy where the inside is slightly negative compared with the outside. Okay. All right. Talking about the actual signaling that goes on, the first type of signals you have are called graded potentials. Graded potentials are caused by some kind of a stimulus. It could be a chemical stimulus, like another neurotransmitter being released. It could be mechanical stimulus. It could be, light rays in the case of your eyes, or sound waves in the case of your ears. But some kind of stimulus is causing the graded potentials to happen. Graded potentials happen on the dendrites and sometimes directly on the cell body, and they involve, either the ligand-gated channels or they involve the mechanically gated channels. And depending on whether you open up the sodium channels or the potassium channels, you're going to create different types of graded potentials. If you open up a sodium channel, what kind of a graded potential do you get? You get a depolarizing graded potential, also called an excitatory graded potential. What if you open up a potassium channel, what kind of a graded potential do you get? You get a hyperpolarizing graded potential, right, where the inside becomes negative, okay? Or it's also called an inhibitory, okay? Remember, when you're opening up sodium channels, it's a depolarizing graded potential, and it's bringing you closer to reaching, what's that magic number we're always asking about? Threshold. It's bringing you closer to reaching threshold. What does threshold mean? Well, first of all, what's the typical value of threshold?
Minus 50.
Minus 55 millivolts. And what's so great about threshold? What happens if you hit threshold?
Action potential.
You get an action potential, all right? Remember, if you perfectly hit it, how many action potentials do you get? 1. If you don't quite hit it, how many action potentials do you get?
0.
None. And if you go above and beyond, how many do you get?
1.
No, if you go above and beyond, you get multiple. Remember? That's how your brain can tell the difference between a weak stimulus and a strong. One of the ways is how fast the action potentials are coming into your brain. Now,
It's like the muscle. Remember, it was like this, and then it goes like this.
Yeah.
well, they are. No, they are all or nothing. You either trigger them or you don't. When you do trigger them, they're always the same size, but that doesn't mean you can't get more than one. Okay, all or nothing just means they always, when they're triggered, they always are the same size. Okay, but with a really strong stimulus, you'll get multiple action potentials versus a weaker stimulus, you might just get one action potential. So it's the frequency of the action potentials that matters, right? And where is the name of the spot on the axon where the action potential begins?
Axon.
Well, it's the axon hillock, but specifically physiologically, it's called the
Initial.
trigger zone. The trigger zone is where it all takes place. Basically, what happens is when you hit negative 55 millivolts, there are voltage-gated sodium channels that are there at that trigger area that are tuned to that number, and that will cause them to open up. Sodium will rush in. That'll then trigger the next voltage-gated sodium channel to open, and then sodium rushes in there and just works its way down. Okay. Speaking of, so again, remember, graded potentials, the size of a graded potential can change based on how strong the stimulus is. A size of an action potential never changes. You either have them or you don't, and that's what all or none means, okay? Other things to know about action potentials, well, sorry, I'm kind of going out of order here. Another thing to know about graded potentials is you can add graded potentials together to get bigger graded potentials. What's that called when you add them together?
Summation.
That's summation, okay? Can you do summation with action potentials?
No.
No, because why? Action potentials have a waiting period called a
Oh,
refractory period, okay? And because of that, you can't basically stimulate another action potential until the other one's completed, so you can't really build on it, okay? Speaking of refractory periods, there are 2 types of refractory periods with action potentials: the absolute refractory period and the relative refractory period. Absolute refractory period means what?
Can't get any more action.
It means no matter how strong the stimulus is, the little ion channels are just not ready yet, and you cannot get another action potential. What does relative refractory period mean?
You can get action if it's stimulus strong enough.
That's right. You can get one, but you're going to have to use a stronger than normal stimulus because things aren't quite back to normal yet. It takes a moment for those gated ion channels to reset themselves, and they're not quite completely reset yet, but they're reset enough that if you have a bigger than normal stimulus, you can get an action potential. Now, when action potentials move down Unmyelinated axons, what's the name of that type of conduction? That's continuous conduction. When they move down myelinated axons, what's that called? Saltatory conduction, which basically means jumping from one node of Ranvier to the next node of Ranvier, et cetera, et cetera. Besides myelination, what other two factors can influence the speed of an action potential?
Temperature.
Size.
So the temperature, higher temperatures is usually faster, colder is slower, and then also
Size.
the size, the diameter. Bigger diameter is faster. 3 classes of axons based on their myelination status, their diameter, and their speed. What's the biggest, fastest fibers called? A fibers. What's the smallest, slowest fibers? C fibers. And then in between you've got the B fibers, right? Encoding the intensity of a stimulus. How does your brain know the difference between a strong stimulus and a weak stimulus? I mentioned one already.
Frequency.
The frequency of the action potentials. So, right, like if somebody touches you lightly on the arm, the nerves carrying the signal up to your brain would be like action potential, action potential, action potential, right? Versus if somebody really forcefully grabbed you, it'd be like action potential, action potential, action potential, action potential. They'd all be the same size, but they would be coming in a higher frequency, right? That's one way. What's the other way that your body can tell the difference? Simply by the number of neurons that get recruited, right? If somebody touches you lightly on the arm, maybe only 4 or 5 neurons are getting involved. If somebody touches you really forcefully, maybe it's 20 or 30 neurons, right? So that's known as neuron recruitment. Now, signal transmission at synapses. What were the 2 categories of synapses that we discussed?
Post and pre. Is that what
No, electrical and
chemical.
yeah, electrical and chemical. Which is faster?
Electrical.
Electrical. Which is slower? Chemical. All right. Every time you have to, when you have a chemical, well, the difference is, remember, with electrical, the cells are physically connected to each other, usually through gap junctions that create passageways. So you don't have to release a chemical to cross some space like you do with a chemical synapse. And because of that, it's faster, okay? But we mostly talked about chemical synapses. That's where you got to release a chemical, it crosses the synaptic cleft, and then it stimulates the next neuron. What's the name for the neuron that comes before the synapse? The presynaptic neuron, and the one that comes after it? Postsynaptic neuron. When you're looking at graded potentials on the postsynaptic neuron, they have special name for it. They call them either EPSPs or IPSPs. What does EPSP stand for? Excitatory postsynaptic potential. What does IPSP stand for? Inhibitory postsynaptic potential. Remember, these are names for things that you already learned. An EPSP is just the specific name for a depolarizing graded potential when you're looking at the postsynaptic neuron, and an IPSP is just really the name for a hyperpolarizing graded potential when you're looking at the postsynaptic neuron. Now, all right, kick this into gear. One thing that's really important is after a neuron gets released into the synaptic cleft, it's going to travel, it's going to stimulate the receptor, but then usually what do you want to do with that neurotransmitter after it's stimulated the receptor? You want to get rid of it so that it doesn't keep stimulating the receptor. What are the 3 methods of getting rid of the neurotransmitter? Enzymes to break it down. Good. Uptake by typically like the neuron, the presynaptic neuron that just released it. And then the third one?
Diffusion.
Yeah, diffusion, just kind of moving out of the way to someplace else. Summation I mentioned before. Summation happens with graded potentials. There's spatial summation. What happens with spatial summation? You've basically got multiple presynaptic neurons and then one postsynaptic neuron. And if they all time it right and at the same time they stimulate that one postsynaptic neuron, you can add all of those graded potentials together. That's spatial summation. Multiple presynaptic neurons simultaneously stimulating the postsynaptic neuron. And then there's temporal summation. Temporal summation is one presynaptic, one postsynaptic, but if the presynaptic rapidly stimulates the postsynaptic, then that can also get the postsynaptic to start adding it together. So Either one of those things, spatial summation or temporal summation, are possibilities. We talked about neurotransmitters, and why is it hard to classify a why is it hard to put neurotransmitters in nice, neat boxes based on their function?
You can do so many in different systems.
Correct. This one neurotransmitter may have different functions in different areas of the body. So like acetylcholine, it might be excitatory in some areas of the body, but then inhibitory in other areas of the body. So you can't put it in a nice, neat box. So it's a lot easier to just classify neurotransmitters based on their chemical structure. And for that, they then basically put it into two groups. You're either a small molecule neurotransmitter, such as acetylcholine or dopamine or serotonin, or you're a large one, in which case they call that a neuropeptide. And those are usually made up of up to 40 or so amino acids that are kind of attached to each other. Endorphins, I'm sure you've heard of those. They're natural pain-relieving chemicals. Encalphins, the mysterious substance P. Those are all examples of neuropeptides. Now, one of the reasons why your body has a lot, you may say, well, why don't we just always have electrical synapses if they're faster than chemical synapses? The reason why you have a lot of chemical synapses is when you release a chemical, yes, it slows things down, but it also introduces modifiability, where you can adjust what's happening at the synapse more than you can, say, if cells were just directly attached through gap junctions, right? So it gives you the ability to modify the effects of a neurotransmitter. 4 ways that were mentioned. Synthesis of a neurotransmitter can be stimulated or inhibited. Release can be blocked or enhanced. Removal can be stimulated or blocked. And then the receptor site can either be activated or blocked. What's the name of like a medication or a chemical that acts like a natural neurotransmitter in your body? What do we call those kind of drugs? Those are called stimulators. No, they're not called stimulators. They're called agonists, right? And then what's the name of like a drug or a medication that blocks the receptor so that the natural neurotransmitter can't attach? Antagonists, right? Or in like drug lingo, they usually just call them blockers. Okay. Neural circuits, remember, neurons don't just sit all by themselves. They like to group with other neurons. When you have one neuron stimulating 2 neurons and then 4 neurons, that's called a diverging circuit. The circuit size is getting bigger. If you have just the opposite, that's called a converging circuit. If you have one where the neurons that are in the sequence give off axon branches that go back and re-stimulate the previous neurons, that's called a reverberating circuit. And then when you have kind of a combination of a diverging and then a converging back again, that's called a parallel after discharge circuit. Are neurons good at mitosis? No, they're bad at it. But neurons have plasticity. What does plasticity mean? They can change, they can grow dendrites or grow axons, and they can make new synapses. And that's what happens when you learn new information or you create memories, right? Now, why don't doing mitosis, actually creating new neurons, that would be referred to as neurogenesis. A couple of reasons why it doesn't happen. Inhibitory influences from neuroglia, absence of the growth chemicals that you had when you're a fetus, rapid scar formation. When you have damage to your central nervous system, scar tissue forms quickly, and that inhibits neurons from doing mitosis. That said, there is evidence of some actual neurogenesis in the hippocampus, so keep that in mind. If you damage a neuron in the central nervous system, it's probably going to die, and so don't do that. If you damage a neuron in the peripheral nervous system, it might be able to repair itself, provided that the neuron cell body is still intact, the Schwann cells are functional, and scar tissue hasn't formed too rapidly. 1 to 2 days, so suppose you cut an axon in the peripheral nervous system. First thing that happens is the Nissl bodies break up. That's called chromatolysis. At days 3 to 5, the axon beyond the injury point and the myelin sheath are going to break down. That's called Wallerian degeneration. After that, macrophages will come in and kind of clean up the area, and then the living Schwann cells will form kind of a tube that directs the axon where to grow, and that's called a regeneration tube. All right, that's all I wanted to say about chapter 12. Chapter 12, as you can see, it's the biggest one that's on it. All right, let me try to cook through these other ones now. So chapter 13, spinal cord, spinal nerves. Again, white matter's on the outside, gray matter's on the inside. What's in the white matter of the spinal cord then? Myelinated axons. And because you're in the central nervous system, they're referred to as tracts, right? If the tract's going up, what kind of information is it carrying? Sensory. And they call those ascending tracts or sensory tracts. If it's going down, it's carrying motor, and that's called a motor tract or a descending tract, right? That's the white matter of the cord. The gray matter is on the inside of the cord. That's a site for integrating postsynaptic potentials, IPSPs, EPSPs. That's basically a spot for summation to take place. Now, in terms of the anatomy of the spinal cord, where does the spinal cord begin?
The foramen magnum.
Foramen magnum, right? So as soon as you're below that hole in the occipital bone, you're technically in the cervical spinal cord. As soon as you go above that hole, you're in the medulla of the brain, right? And where does the spinal cord end? Like the L1, L2 vertebrae level. And what's the name of the structure it kind of forms as it comes to a point? Conus medullaris, right? What protects the spinal cord? Vertebrae, obviously, that's going to be a big protector. And then also what? The meninges, right? What's the outermost meninge? Dura mater, which means, what does it translate into? Tough mother. And then the middle layer is called the what? Arachnoid mater. And in the arachnoid mater, what's the name of the space that's right below the arachnoid mater? Subarachnoid space. And what's inside that space? Cerebrospinal fluid. And then finally, what's the innermost meninge? The pia mater, and it's like directly attached to like the spinal cord, and it's got a lot of important little blood vessels in it. Another thing to know about the pia mater is it helps to form anchors that kind of hold the spinal cord in place. What's the name of the anchors that kind of hold the spinal cord to the sides?
So start with the C.
No.
Is that no?
It starts with a D. Rhymes with the
Oh,
denticulate.
Yeah,
The denticulate ligaments. Okay. So they hold the cord to the side. And then what's the thing that's like a long kind of rope anchor at the end of the spinal cord that holds it to the coccyx?
Phylum something. Terminate.
Yeah, phylum terminale. Yep, the phylum terminale. All right, good. If you look at the width of the spinal cord going down from cervical to, down to lumbar, and does it stay the same thickness the whole way?
No.
No. Where is it thicker?
On the
Cervical.
Cervical region it's thicker, and where else? Lumbar. Why is it bigger in those areas? Extra neurons coming in from your upper extremity for the cervical enlargement, and extra neurons coming in from your lower extremity for the lumbar enlargement, right? All right. Other stuff to say. So, remember that the spinal nerves are connected to the spinal cord using roots. What kind of neurons do you find in the dorsal root? Sensory. What kind of neurons do you find in the ventral root? Motor, right? How can you tell the dorsal root from the ventral root? Just looking at it.
Dorsal root ganglia.
Yeah, there's kind of a bulge called the dorsal root ganglia. And again, what's inside that bulge?
The body.
It's the cell bodies of incoming sensory neurons, right? All right. Spinal nerves. Again, spinal nerve, it's part of your peripheral nervous system. Now, in order to hold nerves together, you've got different layers of connective tissue. What's the layer of connective tissue called that surrounds each individual axon?
Exoloma.
That's called the endoneurium,
Oh.
okay? Now you put a bunch of axons together, you get something called a fascicle. What holds the fascicle together? Perineurium. And then if you put all the fascicles together, you get the entire nerve, and what holds that together? The epineurium, right? Remember, that was the same prefixes that you used for muscle, right? The epi and the endo and all that stuff. All right. How many pairs of spinal nerves are there? 31 pairs. All of the spinal nerves, except for C1, come out through holes on the sides of vertebrae when the vertebrae are stacked. What's the name for those holes on the sides? The intervertebral foramen, right? So inter means between vertebrae. So C1 is actually coming out in between the occipital bone and the first cervical vertebrae. So by definition, it's not coming out through an intervertebral foramen. Now, as soon as you have these spinal nerves, they immediately give off branches called rami. What's the biggest ramus that you have? The anterior ramus, and that's because it's basically supplying sensory and motor to the sides of your body, the front of your body, your entire upper extremity, your entire lower extremity, versus the posterior ramus is a lot smaller. It's basically just going to your back region and doing sensory motor on your back region. There's a branch called the meningeal branch that goes back in and supplies the vertebrae and meninges and spinal cord blood vessels. And then there's the rami communicantes, which we'll talk more about later, but they're basically branches of your sympathetic nervous system, which is part of the autonomic. Now, to make your life miserable, most of the anterior rami, after they come out, they like to interconnect with other anterior rami, either above or below them, to kind of help organize the neurons before they go to different areas of the body. What's that name of those interconnections you have between the anterior rami?
Plexus.
Plexuses. True or false? You have a plexus everywhere in your spinal cord.
No.
False, right? Where's the spot where you don't have a plexus?
Thoracic.
Thoracic area, okay? Everywhere else, you pretty much have plexuses. What's the highest plexus you have?
Cervical.
Cervical plexus. And what is the famous cranial nerve that comes off of the cervical plexus?
Phrenic.
The phrenic nerve. And why is it famous?
Because
It keeps you alive. It goes to the diaphragm, right? And that's your main breathing muscle. What's the next plexus you have after the cervical plexus? Brachial plexus. And that's the plexus that organizes the nerves that are going to go to your upper extremity, right? Then you go through the thoracic area. You get a break. There's no plexus in the thoracic area. Then you've got the lumbar plexus. Lumbar plexus is going to your lower limbs. The largest nerve from the lumbar plexus is the femoral nerve. Then after that, you go into the sacral plexus. Sacral plexus is famous for giving rise to the largest nerve in the body, which is the sciatic nerve. And then finally, you have a little tiny coccygeal plexus. Next, we talked about the concept of dermatomes. Dermatomes are areas of the skin that get supplied with their sensory by particular spinal cord levels, okay? Remember, or by particular spinal nerves. Remember, every spinal nerve has a dermatome except for C1, okay? And then also remember that of your cranial nerves, one of your cranial nerves has a dermatome. Which of your cranial nerves has a dermatome?
Number 5.
5. Cranial nerve number 5, which is our friend, the trigeminal. If you want to write the word dermatome over by where it says sensation to the face, it's the cranial nerve that has a dermatome. Okay. All right. Now we talked about tracts. Tracts, again, are bundles of axons within the central nervous system. Sensory tracts go up, motor tracts come down. Tracts are usually named, or often named, based on where the tract begins and where it ends. And if you know that, you can then kind of figure out what kind of tract it is. So the corticospinal tracts, where do they start?
Brain.
Specifically, what part of the brain?
Cortex.
Cerebral cortex, that's what the cortico means. And then where do they end?
Spinal.
Spinal cord. So are they sensory or motor? Motor, right? They're starting up high, and then they're ending low, so they're going downward, so they're motor, right? Spinal thalamic tract, where does that start? Spinal cord, where does it end? Thalamus. So it's starting low, it's going up, so it has to be sensory, right? Let's talk about these tracts, though, in a little bit more detail, starting with the sensory tract. So I mentioned the spinal thalamic tract. What is the spinal thalamic tract most famous for? What sensation? Pain, okay? It also happens to do temperature. It does itch, tickle, deep pressure, crude touch, but it's most famous for doing pain. By the way, it's also known as the anterolateral tract. If you ever see that, spinal thalamic and anterolateral are the same thing, okay? Another sensory set of tracts you have, are called the posterior column tracts, and there's actually two that make it up, the Grassoff fasciculus and the cuneate fasciculus. They do discriminative touch, light pressure, vibration, and then conscious proprioception. What's conscious proprioception? Knowing where what is?
Your
Yeah, kind of
body
knowing where
is-
your body parts are in space without having to look. Like, you know, if you do this and you close your eyes, you know your arms are out in front of you. You don't like freak out like, oh my God, where are my arms? Where are my arms? Oh God, okay, they're still in front of you. You know they are because of the muscle spindles, the Golgi tendon organs, the joint kinesthetic receptors. They're telling you, they're giving you that sense of conscious proprioception. So that's carried by the Grassoff fasciculus and the cuneate fasciculus as well. So that's sensory. The motor tracts, they like to put them into two categories called the direct pathways and the indirect pathways. Direct pathways do voluntary movements. Indirect pathways do automatic movements. Now, they're both going to skeletal muscles, but direct are doing things that you're more in voluntary control of. Indirect are doing things like, you know, swinging your arms automatically when you walk, okay? Something you don't really think about, but you do it anyway. Keep in mind for the direct pathways, they all start up really high in the cerebral cortex, and then they go down and they end in a lower place. And there are two that go and control your body, your arms and your legs and your trunk. They're called the corticospinal tracts. There's a lateral and anterior corticospinal tract. And then there's another direct pathway called the corticobulbar that actually starts up high in the cerebral cortex. Bulbar is a word that refers to the brainstem. So it's starting in the cerebral cortex, it's going down in the brainstem, and that's actually going to synapse with your facial nerve, and that's going to do voluntary movements of your face. Okay, so the corticobulbar tract is really important for that. Indirect pathways, they start lower in the brain. They start not in the cerebral cortex. They start someplace in the brainstem, and then they usually go down and they end in the spinal cord. Wherever they start in the brainstem is going to be the first prefix of the name of the tract. So you have lots of crazy names such as rubrospinal. Okay? Anyone know what rubro means? Red. That's going to start in the red nucleus of your midbrain, and then it's going to go down and it's going to end in your spinal cord. Or the tectospinal. You already know, hopefully you remember, the posterior part of your midbrain is called the tectum. That's where this tract starts. It starts in the tectum, and then it goes down your spinal cord. Vestibulospinal, that's going to start in the vestibular area that's located within the brainstem, and then go down. And then there's a reticulospinal that starts kind of in that reticular formation area that also extends throughout the brainstem, and then it goes down. Again, indirect pathways, they're doing more automatic movements, things that you don't have to think of. Lastly, we finished with reflexes. Reflexes are basically negative feedback systems. They're monitoring some body condition, and if it falls outside its normal range, they're trying to quickly fix it and put it back in its normal range. 5 parts to a reflex arc. What's the first part of a reflex arc? Sensory receptor. So it's monitoring the body condition. What's the second part of a reflex arc?
Nerve.
The sensory neuron, right? That's connected to the sensory receptor. What's the third part?
Interneuron.
Yeah, or like the integration center. Now there may be an interneuron there, or there may not be, but it's kind of the control center for the whole thing. What's the fourth part? The motor neuron that's going out. And then the 5th part is the effector. And again, if it's a somatic reflex, the effector is always what? Skeletal muscle. If it's an autonomic reflex, it's smooth muscle, cardiac muscle, or glands, right? Remember, there's lots of ways to classify reflexes. If the sensory signal is on the same side coming in that the motor signal goes out, what do you call that kind of a reflex? Ipsilateral. If they're on opposite sides, you call it contralateral. Which most reflexes that we studied were which one? Ipsilateral, right? Usually the sensory and the motor were on the same side. If you have an interneuron between the sensory and the motor neuron, what kind of a reflex is that? That's polysynaptic reflex, versus if you have the sensory directly synapsing with the motor, you call that a monosynaptic reflex. Which is faster? Mono, right? Every time you introduce a synapse, it's going to slow things down, okay? Stretch reflex, tendon reflex, flexor withdrawal reflex, those were all ipsilateral reflexes. What was the one that we studied that was a contralateral reflex?
Cross.
The crossed extensor reflex, and that was designed to help you do what?
Balance.
Maintain your balance when you step on something, say, painful with the opposite foot. This one is causing the opposite leg to basically extend to support your body weight so you don't fall over, right? Also remember, most reflexes have reciprocal innervation. What does reciprocal innervation mean?
Antagonist.
It would innervate something as a response to activating something else.
Yeah, it's basically a coordination between two muscles. You're going to want to tell one muscle to do one thing, but then you got to make sure that the antagonistic muscle is doing the opposite, or else the reflex won't work. So when you do the knee-jerk reflex, you want to tell the quad muscle to what? To contract, right? But at the same time, you also want to do what?
Relax.
Tell the hamstring to relax, or else the reflex won't work, right? That's reciprocal innervation. All right, chapter 14, brain and cranial nerves. What are the four major parts of the brain? So, what'd you say? Cerebrum, cerebellum, brainstem, diencephalon. Thank you. What are the three parts of the brainstem? Midbrain, pons, and medulla. Cerebellum, there's really just one part to that. What are the three parts to the diencephalon? Thalamus, hypothalamus, and epithalamus. And then there's really just like one part to the cerebrum as well. I mean, aside from the lobes, but we'll get into that later. Same protection that you had for the brain, you have with the spinal cord. Blood flow to the brain. Is blood flow to the brain? Is there good blood flow?
Yes.
Oh yeah. There's two pairs of arteries that supply fresh oxygenated blood to the brain, the internal carotids and the vertebral arteries. And then you have these big kind of collecting veins called the dural venous sinuses that take the deoxygenated blood away and they dump it into your jugular veins. What's the name of the thing that makes it more difficult for potentially harmful substances in your blood to get into your brain? The blood-brain barrier. The blood-brain barrier is made up of tight junctions between the endothelial cells that make up the wall of the blood capillaries, an extra thick basement membrane, and then finally the astrocytes. They have these projections that make it as well. CSF, cerebral spinal fluid, is a clear fluid that circulates around the inside of your brain and spinal cord and then circulates around the outside in the subarachnoid space. In terms of the cavities that are inside your brain, what's the name for the cavities that are inside your brain? The ventricles. You have, inside the cerebral hemispheres, you have the two what? Lateral ventricles. Let's go through this. So let's suppose you're some CSF and you've just been made in the lateral ventricles. Again, what's the name of the glial cells that make CSF? Ependymal cells. And what's the name of the little spots where the CSF is actually made? Choroid plexuses. And basically, what are you doing when you make CSF? You're just what? You're basically filtering the blood. You're taking like the liquid part of the blood out, okay, and you're filtering it. So you make some CSF, you're in the lateral ventricles. Where does the CSF go next?
Aqueduct?
No.
Third ventricle.
Before that, interventricular foramen. Then it ends up in the third ventricle. Then where do you go?
The cerebral aqueduct.
Cerebral aqueduct. Then you're in the fourth ventricle. Then you have some options here. If you want to stay within the CNS, you could then continue down in the central canal, right, of the spinal cord. Or you could also go out through yeah, you will go out through, what's the name of the holes? The apertures, the lateral and the median apertures. And then you're going to be in the subarachnoid space. You're going to circulate around the outside of the brain and spinal cord until, and then this was the quiz question, where do you return it back to the blood? Arachnoid villi. Okay. Good. Looks good. What's the name of the problem when you have like a blockage and you have buildup of CSF? Hydrocephalus. Where does the blockage usually occur?
The aqueduct?
Yeah, the aqueductus sylvius is the most common place. All right, we talked about the brainstem first. That makes up the medulla, the pons, and the midbrain. On a scale of 1 to 10, 1 being not important, 10 being important, where would you rank the brainstem? 10, 11 probably. It's the most important part of your brain in terms of just keeping you alive because it has lots of important vital centers. Okay, the vital centers are basically different nuclei that are going to be in the brainstem. Remember, the definition of a nucleus is a cluster or a collection of neuron cell bodies in the central nervous system. And in the brainstem, these nuclei are there to support different vital functions. So there's a cardiovascular center that controls your heart, a medullary rhythmicity center that controls your breathing. There's also centers, there's a vomiting center, a swallowing center, a whole bunch of things that are located inside there. So that's all within the medulla. If you go up from the medulla, what's the area that you're going to be in after the medulla? The pons. The word pons literally means bridge. It's a connection spot between different areas of the brain, sending signals from, like the cerebrum, for example, then sending them into the cerebellum, or sending them from like your lower extremities and then bringing it into your cerebellum. Okay, so it's a connection point for that. The pons also has a vital center in it for breathing. Something called the pneumotaxic and the apneustic areas. Those are important. They help out the medullary rhythmicity area for breathing. Just above the pons, then you have the last part, which is the midbrain. What's the posterior part of the midbrain called? You can call it the tectum, or specifically the name for the 4 bumps are the corpora quadrigeminum. The 2 upper bumps are called the what? Superior colliculi. And what do they control? Head movement, body movement in response to what kind of stimuli?
Audio. No.
Visual. Superior are basically
Visual.
turning your body, your head, your eyes in response to some visual stimuli. And then the inferior do the same thing in response to
Visual.
Sound.
No.
auditory.
I'm confused.
Do not listen to anything she just said. She was 100% wrong. Yeah. Superior is visual, inferior is audio. Okay? Inferior is audio. All right, let's see here. Other areas of the midbrain, the substantia nigra, that's a place where the neurons there release dopamine. It's important for subconscious muscle activities. It's a problem that, it's an area where neurons are lost in people with Parkinson's disease. I mentioned the red nucleus is also there. A couple of things. Now, the reticular formation is an area that extends throughout all 3 parts of the brainstem. The word reticular means net-like, and it's because it's a combination of myelinated axons and neuron cell bodies that give it, I don't know, what looked like a net to someone. They thought it looked like a net. The most important part of the reticular formation is the ascending part that they refer to as the reticular activating system. 3 important functions of the reticular activating system.
Association.
So maintain consciousness, what else?
Keeping away.
Wake you up from sleep, and then filtering out extra information in the environment so you're not overloaded, like preventing sensory overload. Okay? All right. Cerebellum, what does the word cerebellum translate into?
Little brain.
Little brain. It's the 2nd largest part of the brain. Again, what's the name of the little worm thing on the back?
Vermis.
The vermis. The 2 sides are called the cerebellar hemispheres. The outer part is going to be gray matter called the cerebellar cortex. Again, what's the name for the white matter on the inside? The arbor vitae. And what's the function of the cerebellum?
The quality of the signal.
Kind of like a quality control
Control
center
and
for your body
balance.
movements. It corrects any errors in your movements. It smooths out the movements. It also regulates posture and balance. What was that name for that problem when you have like poor coordination and ataxia, right? Okay, let's see here. Diencephalon comes next. Just remember there's 3 parts to it: thalamus, hypothalamus, and epithalamus. Which of the 3 parts is the biggest? Thalamus. 80% of the diencephalon is the thalamus. And the thalamus, what's the function of the thalamus? It's kind of like a relay center for incoming sensory signals, okay? Remember, we'll talk about this more, I think in chapter 16, but most sensory signals, as they're going up your spinal cord and using those different tracks that we talked about, they eventually have to reach your cerebral cortex for you to be consciously aware of them. But most of them don't go straight to the cerebral cortex. They stop off and they synapse with another set of neurons at the thalamus. So It's a stopping off point or a relay station for most sensory neurons, with the exception of what sensation? Smell. Smell goes straight to your cerebral cortex. It doesn't stop off at the thalamus. Okay. Let's see here. Hypothalamus is a relatively small part. It's important for, when you hear hypothalamus, think of the word homeostasis. They both start with the letter H. It's a major regulator of homeostasis. There's a lot of important sort of control centers for homeostasis located in the hypothalamus. For example, there's eating and drinking centers, and there's feeding and satiety centers, a thirst center. There's also the control center for your body's temperature located there. So a couple of important things going on in there. And then the epithalamus. What's the name of the gland that you have in the epithalamus?
Pineal.
Pineal gland. And what does it produce? Melatonin. When during a 24-hour period are melatonin levels highest in your blood?
3 a.m. Oh.
Yeah, I mean it's around there. It's don't quote me on that, but it's sometime like usually when it's really dark, or usually early in the morning around 3 a.m. something like that. And what does it do to you? What's it supposed to do to you? Calm you down, make you feel sleepy, etc. All right, cerebrum. Cerebrum is the largest part of your brain, and the outer part is the cerebral cortex. It's going to be gray matter. It develops faster than the underlying white matter, which causes those folds. What's the name of the folds that you have on the cerebrum? Gyri, or the singular form is gyrus. When you have a deep groove, they'll call it a fissure. When it's a more shallow groove, they call it a sulcus. What's the name of the fissure that divides the cerebrum into the right and left hemispheres? Longitudinal fissure. What's the name of the big sulcus that kind of cuts in a coronal fashion? That's the central sulcus. If you're immediately, okay, let's talk about now the lobes then. What are the four main lobes of the cerebrum? Frontal lobe, parietal, occipital, and temporal. Okay. Just keep in mind, those are the four main ones. There is a fifth one. What was the fancy fifth one? The insula. Okay. But the ones they usually talk about are the other four. Now remember that the two cerebellar hemispheres, they're not totally separate from each other. There is a big connection spot between the two of them. What's that big connection spot where axons cross sides? Corpus callosum, right? That's a big spot where they connect. If you look inside after the cerebral cortex, you're going to have mostly then white matter, which represent myelinated axons in different types of tracts. Tracts that connect different gyri within the same hemisphere are called what kind of tracts? Association tracts. Tracts that cross sides and connect gyri in different hemispheres are called commissural tracts. And the corpus callosum basically has the biggest commissural tract. And then tracts that go up or go down are called projection tracts. Okay. If you go really deep inside your brain, you do encounter more gray matter. That's called the basal nuclei. They help initiate and terminate movements. They suppress unwanted movements. They regulate muscle tone. So they do a lot of important things. All right. Functional organization of the cerebral cortex. If you're in the frontal lobe, what kind of function in general does the frontal lobe have? Motor. If you're in the parietal, the temporal, occipital, what do they have? Sensory, and then also association. Okay, that's kind of the general big picture. If you go immediately posterior to the central sulcus, what's that gyrus that you're in? Well, immediately posterior, you're in the post-central gyrus, and that's where you find the primary somatosensory area. It's part of your parietal lobe, and that's where, like, skin sensations have to reach for you to become consciously aware of them. Okay? What if you go immediately anterior to the central sulcus? You're in the precentral gyrus, also called the primary motor area, and that's a part of your frontal lobe, and that's where your ideas for voluntary movements originate. Okay? Remember, with both of those areas, they can do that little, what's the name of that little man that you put on the brain? the homunculus, right? And remember that the size of the body part is not necessarily representative of its actual physical size. Instead, it's based on, in the case of the sensory homunculus, how sensitive that body part is, right? And in the case of the motor homunculus, how much fine motor control you have over that body part, right? Broca's area. What's Broca's area?
Speech.
It's a motor speech area. It's in the frontal lobe, close to the lateral cerebral sulcus, and it basically is going and it's controlling the muscles that cause you to speak properly. And if you have damage in Broca's area, what's the name of that problem people get? That's called a Broca's aphasia or a non-fluent aphasia. And what do they sound like?
Word sound.
You can't tell, right? They're just, they're like mumbling the words. You don't know what they're saying. Association areas, remember, these are areas that usually sit close to the primary sensory areas, and they give meaning to the incoming sensation. So to become consciously aware of a sensation, it first hits a primary sensory area. But then very quickly, a connection will be made to an association area, and that'll like attach meaning to that particular sensation that's coming in. Okay. Hemispheric lateralization, just remember, even though they anatomically look like kind of mirror images of each other, usually on most of us, we're either right brain or left brain dominant. Who's usually more lateralized, men or women? Men are usually more lateralized. They kind of put all their eggs in one basket, right? Whereas women kind of spread out the function between the right and the left hemispheres. Brain waves, what's the name of the test you can do to test someone's brain waves? Yeah, an EEG, an electroencephalogram. 4 types of brain waves. Alpha waves are like your standard waves you see when you're awake. Beta waves are what you see when you're like really thinking. Theta waves are when you're stressed out emotionally or brain disorder, and then delta waves, you see those when you're in deep sleep. If you see them in someone that's awake, it usually indicates severe damage. Cranial nerves, we did go through all this stuff just very quickly. I mean, make sure you know this table really well. Make sure you know, definitely that classification thing. If you need to resort to this thing, don't be shy. You can resort to that thing. Make sure you know which guys, I didn't actually summarize it, but make sure you know the guys that are PSF, or that are parasympathetic, right? 3, 7, 9, and 10, right? Oculomotor, facial, glossopharyngeal, and vagus. 3, 7, 9, and 10 are the ones that have parasympathetic branches to them. Yeah, so just make sure you go over this really well. I don't have anything in particular to point out. Just make sure you know it very well. Development of the nervous system. Remember, it's all about the ectoderm. What's the name of that flat thing that you have that should fold?
Neuron.
That's the neural plate. And when it folds, it's called the neural tube. Disorders of the brain. We talked about CVAs or strokes, right? What are the 2 categories of strokes? Ischemic and hemorrhagic, okay? Abrupt onset, paralysis usually. Again, remember with a stroke, if you have a stroke on the left side of your brain, what side of your body is usually paralyzed?
Right.
The right side. So usually the symptomology is contralateral to where the pathology is, right? TIA, what does TIA mean? Transient ischemic attack. It's like not permanent brain damage. Symptoms go away, but you should always take a person in. Alzheimer's disease, 11% of the population, debilitating, progressive, etc. All right, that's it. I somehow made it through all that stuff. Any questions about any of that stuff? All right, well, good luck. If you think of anything,